everyone. Let's begin with today's class on Enzyme Science and Technology. So today's class will study more about enzyme application in different fields like food industry, medical field, and diagnostic field. So enzyme has different application. It has application in various fields like agriculture, poultry, in vaccine preparation, also in preparation of transgenic animal, and in many therapeutic applications like enzyme is used. Like we studied an example in the previous class where different inhibitors were used, enzyme inhibitors were used to inhibit acetylcholinesterase enzyme, which is involved in muscle contraction and relaxation and other activities. So enzyme has many roles in therapeutic fields as well, and we can also so enzymes are also used in making genetically modified organisms, and in other processes like drug delivery, and also in preparing transgenic crops in agriculture. So these are the different applications of enzyme, and we'll study mostly about these four applications of enzyme. That is when enzyme is involved in food industry, medical field, genetic engineering, and in environmental processes. So let's begin with the food industry first. So for the food productions, this is the overall schematic of food production. For example, here we have milk production. First we have in the first step of the milk production, we will have the animals which we get milk from, like cows and other animals, and then that milk which we have got is processed in the industry. It is processed for like it can be pasteurized, and other processing are done in order to increase the shelf life of the milk. All these processes occur in the processing step, and in this step there are many enzymes are involved during each of these processes. And then after processing the milk, it is further packaged for distribution, and then we can distribute it to the retail or directly to the consumers at restaurant level, or at retail level, where all the common people can buy from different retailers, and then it is finally distributed to the consumers. Either it could be home consumers or the restaurant consumers. So finally reaching to the consumers, it takes a lot of time, and that lot of time the processing industry takes care of the different methods in order to ensure the milk has sustained the proper nutrients, and all the nutritional value is retained, and also these processes help in retaining the shelf life of the milk. So let's understand what are these processes and which enzymes are involved in this food processing industry. So in dairy production, it's a very huge industry, and there are different kinds of enzymes used, like rennet, lactate, protease, and catalase. Then we have brewing industry. So in brewing industry is nothing but where alcohol and beer is made, and we use different enzymes like beta glucanase, alpha amylase, protease, and amyloglucosidase. So this is also a huge industry, and these are the enzymes, major enzymes which are used in the brewing industry. And in the baking industry, we use maltogenic enzyme like maltogenic amylase. These are mainly carbohydrate degrading or reducing enzymes, glucose oxidase, and pentonases. Then we have wine and fruit juice industry, where we use plant cell degrading enzymes like pectinases and beta glucanases. And then finally we'll also learn about meat industry, where the meat is tenderized and softened by using major enzymes like proteases and taurine. So these are the major food producing industries. So let's begin with the first industry, that is dairy production. And in dairy production we have the enzyme rennet. So this is very important enzyme. Okay, rennet is very important enzyme. It is isolated from fourth stomach of young calf. So cows have 4 different stomachs, and this rennet enzyme is derived from an animal source, and it is isolated from the fourth stomach of the young calf. Okay, and sometimes rennet is also artificially synthesized and used for the dairy production. So what does rennet do? Rennet converts milk. It essentially converts milk to cheese. Okay, so rennet will so we have milk which has it will basically curdle the milk. Rennet will curdle the milk. Okay, and it will separate solid curd and liquid whey. So it will separate this solid curd and liquid whey, and this solid curd is further processed to get cheese. So this is why rennet is very important, and rennet is also isolated from a vegetarian source, and that rennet is known as theistic. Okay, so this is the first enzyme, rennet, which is used in the dairy. The second enzyme is the lactase. So lactase is essentially isolated from yeast. And lactase, as the name suggests, it will break down lactose to glucose and galactose. Okay, so lactose is a disaccharide made up of glucose and galactose, and lactase enzyme, it will break down lactose into glucose and galactose. So this lactase, why is it used so largely in the dairy industry? Because many of the population, as you might have already know, they are lactose intolerant, and this lactose is present in the large amount in the dairy products like milk, cheese, condensed milk, ice creams, and other milk products. So these lactose intolerant people, they cannot consume these dairy products. Hence, for them, these dairy products are treated with lactase enzyme, and this lactase will break down all the lactose which is present in the dairy, and thus it will not affect the lactose intolerant people. So this is so lactase is that's why very largely used in the dairy productions, because in order to completely digest or completely degrade the lactose content in the milk. The third enzyme is protease. So protease is also they will degrade proteins. Okay, so during cheese production, these proteases are used in order to enhance the flavor of the cheese. So hence proteases they enhance a particular flavor give particular flavor to the cheese, which is the characteristic of that type of cheese. So that's why proteases are used. And finally catalase. So catalase again it's isolated from bovine liver. Okay, it's isolated from bovine liver, and also it is isolated from microbial source. So we have different microbial source in which it is isolated. So catalase enzyme, what it will do, catalase breaks down H2O2 into H2O plus oxygen. Okay, oxygen gas. So catalase will break down H2O2. Okay, so many H2O2 is widely used. So H2O2 is very harmful, and it is widely used to protect the food and the wrappers from oxidation. So and H2O2 is also used for the cold sterilization processes. So in order to remove H2O2 from the food, we use catalase enzyme. So proteases, as I mentioned, there are different types of proteases. So there are exopeptidases and endopeptidases. So what are these types of proteases? They will either so suppose this is our protein of our interest. So the proteases can chop off the protein on the end. If they chop off from the ends, they are known as exoproteases. Okay, and if they chop off in between randomly, they are known as endopeptidases. So depending upon the site of the action, if the site of action is at the end, either at the N terminal or C terminal, then either from N or C terminal, then they are exopeptidases, or endopeptidases can chop in between. Another type of proteases are so they can be divided into serine, cysteine, aspartic and metalloproteases. So these are the amino acids which are present in the active site of these proteases. So that's why they are known as serine proteases, cysteine proteases. So the serine is the functional group. Serine has a OH functional group, and this OH is responsible for the catalytic activity, and that's why the name serine, cysteine, aspartic and metalloproteases. Similarly, in metalloproteases there will be some metal ion which is actually contributing to the catalytic activity. And then we have pH. Then we have pH dependent proteases like acid, alkaline and neutral proteases. So these proteases act at particular lower or higher pH, and that's why their classification is based upon the pH in which they actually catalyze the activity. So proteases, as I mentioned, can be from either so proteases are mostly isolated from microbial sources like bacteria. So as you can see here, we have alkaline proteases isolated from Bacillus species, and neutral proteases are isolated from Pseudomonas species. So depending upon the application, whether we want alkaline proteases or neutral proteases, that will depend upon the type of food and what is the pH or what is the application which we want to use. If we have If you want the activity at a very high pH, that is in alkaline condition, we use alkaline proteases. And similarly, if you want activity at neutral pH, then we use neutral proteases. And these are the different industries in which the proteases are used. In detergent, leather, food, neutral proteases are used in food industries. Then proteases are also isolated from fungi, aspergillus species. So these aspergillus species, they can also give alkaline or neutral proteases. So we can isolate both alkaline and neutral proteases from aspergillus species, and we have mucor and endothea parasitica species also. That will give us acidic proteases. And these are the different species which are used for the isolation of proteases. And these isolation, we can isolate these proteases by using different chromatographic and other techniques, okay, which we have already studied. Okay, these are the techniques which are used for the purification of these enzymes, and we've studied this in our previous classes. So let's move on to brewing industry. So brewing industry is very major industry, and so these are the different enzymes which are used in the brewing industry. That is protease, beta glucanases, alpha amylase, and amyloglucosidases. So proteases again, it will break down. Proteases again will break down protein into amino acids, right? And these breakdown of protein will give a specific taste, specific bitter taste to the alcohol, to the brew. Okay, and this taste is the characteristic taste of the whatever brew which is brewing. Okay, so proteases are also important. It has different application. That is, these amino acids which are formed here. These are the amino acids that can be source of energy for the yeast that will convert alcohol. Okay, the yeast that will convert alcohol, that will increase the alcohol content. And also proteases reduces haze during brewing. So these are the different application. Okay, amino acids from the degraded protein can be a carbon source for yeast, and it will convert more alcohol from those amino acids. And also proteases are used to reduce the haze in the brewing industry. Then we have beta glucanases. Beta glucanases will break down beta glycosidic linkage. And this beta glycosidic linkages are present in carbohydrates that will again aid in mashing. Okay, it will help in mashing and brewing. Okay, it will help in mashing and brewing, and it will also be the source of carbon for the yeast. Then we have alpha amylases. Alpha amylases will convert starch to dextrin. Okay, and this again will be source of carbon for yeast. It solubilizes the carbohydrates, and it decreases the mashing time. So that is why alpha amylases are used to break down the starch and convert it into dextrin, and this dextrin can be further converted to alcohol, ethanol. So alpha amylases are isolated from various alpha amylase again is very important. It is isolated from Bacillus species and also different fungi species like Aspergillus and Mucor species. Okay, also from yeast like Candida and Saccharomyces. And also thermophilic bacteria. So, what are the application of amylases? We'll quickly write down few points on the application of alpha amylases in the brewing industry. So the main objective of to use amylases is starch degradation or hydrolysis. Okay, so then it will convert in for beer formation or for alcohol production, and then we also so this is for alcohol production. And also it can be used for extracting fruit juices from the fruits. Then amylases also have anti-stalling effect. Okay, it has anti-stalling effect. Alpha amylases are anti-stalling agents, and they are used again in mashing and brewing. So these are the application of amylases. So the next food industry is baking industry. So in the baking industry, instead of alpha amylases, we use maltogenic amylase. Okay, again the maltogenic amylase is it will break down the starch, right? It will break down starch hydrolysis. Okay, and this starch hydrolysis will be carbon source for yeast. Again, it's an anti-stalling agent. And the third application, it also converts starch to gelatin. Okay, so gelatinizes the starch. Then we have glucose oxidases. So basically glucose oxidases will convert glucose to gluconic acid. Okay, it will oxidize the glucose and it will convert gluconic acid and H2O2. So this H2O2 it will strengthen the disulfide cross-linking between the enzymes. Okay, and then we have pentonases also. So these pentonases are used for the dough mashability. Which makes it very easy to handle the dough. Then it stabilizes stabilizes the dough. Now, in the food industry, the final industry is wine and fruit industries. So, in the wine and fruit industry, we have pectinases and beta-glucanases. So, what are these pectinases? So, pectinases are, it will basically prevent haze formation because of pectins. Pectin hydrolysis. And it also helps in extraction of color and color from fruit juices. So the outer covering of grapes or any other fruit juices, they are rich in pectinases. They are rich in pectin. The cell wall of these fruits are rich in pectin. So pectinases are used to extract the color from the fruit in fruit juices and also in the wine industry. Okay, and pectinases are also used to break down protein breakdown. And it removes mucilage during these processes, and it promotes antioxidants in the juice, and also removal of inner wall of the seeds. So that we can extract juice and all the contents from the seeds or the fruit. So the sources of pectinases. So what are the sources where we can isolate? We can isolate pectinases from filamentous fungi. And we can isolate from vegetables like tomatoes, oranges, and Bacterium Clostridium species. So these are the sources for the pectinases. The next enzyme is after pectinases is beta-glucanases. So beta-glucanases also it reduces maturation of fruits. And also it helps in, it improves the action of other enzymes. So now let's move on to meat industry. In the meat industry, we majorly use two enzymes. First is papain. Papain is isolated from papaya. can be isolated in form of a crude mixture, like we can use just papaya pulp, crude papaya pulp, or it could be in solid powder which contains the enzyme peptide. So this peptide is used for softening the meat, softening and tenderization of meat. And this will lead to very soft and tender meat. And the another enzymes which we use are proteases. So again, we studied in the last few slides back that there are different kinds of proteases: exoprotease, endoproteases, and serine, cysteine, threonine proteases based upon the active site of the functional group active site. And then we have different acidic, alkaline, and neutral proteases, and they are sources from different microorganisms. Okay, so these are the different proteases which are used. So these proteases will also soften and tenderize the meat, and after softening the these meat are packed and are further processed. Now next move on to the medical field. So we have completed the food industry, which included dairy, brewing, baking industry, wine and fruit juice industry, and these comprise of the food industry. Now let's move on to medical industry. So in medical industry there are different types of enzymes which could be used. So which could be used either as a diagnostic marker. Okay, they could be either a diagnostic marker or they could be used as a therapeutic application. So these are the different enzymes like aminotransferases, alanine transaminases, also known as ALT, and it also ALT is also known as serum glutamate pyruvate aminotransferases, also known as SGPT. So there are tests like SGPT and SGOT, and these refer to these two enzymes, alanine transaminases and aspartate aminotransferases. So aspartate aminotransferases, also known as SGOT, which is referred to as serum glutamate oxaloacetate aminotransferases. So SGPT and SGOT are used for both liver to check the as a diagnostic marker for liver functioning and heart functioning. And then we have lactate dehydrogenases. This enzyme is also used very widely. And then we have creatine kinase, also known as creatine phosphokinase or CPK. And then we have gamma glutamine transpeptidase GGT. And there are other enzymes which are used at different in different clinical situations and diseases like diabetes and Alzheimer or other diseases. So these enzymes are used for the rapid diagnosis. So whenever there is any bodily dysfunction, or whenever there is some kidney dysfunction or liver dysfunction, we look for these enzymes. Why? Because whenever there is some organ dysfunction, what will happen? It will lead to necrosis or tissue damage. That will lead to release of these enzymes in the blood, And when we give our blood for analysis, these enzymes can be increased at their so there are levels, normal levels, which these enzymes are present in the blood, and if they are found to be increased, if the levels are found to be increased, then we can say that the organ, whichever organ, for example, if the kidney is not functional, then there could be some necrosis happening in the kidney, and there is some dysfunction. The kidney is not properly functioning and not filtering out the urine, so there will be more creatine kinase present in the blood, which is the direct marker for kidney dysfunction. So after doing blood analysis, we can check the creatine kinase level or CPK levels. If they are found higher than the normal, then we can say the kidney is not properly functioning or there is something wrong with the organ. So this is how different diagnostic markers, these different enzymes are used as a diagnostic markers for various diseases and conditions. So let's move on to alkaline phosphatase. So alkaline phosphatase, as the name suggests, there are different isoforms of alkaline phosphatase. Okay, these hydrolyze many types of phosphate esters. Okay, so these alkaline phosphatase are present in both humans and animals. The major source of alkaline phosphatase are present in liver, bone, kidney, and placenta. So whenever there is some dysfunction in the liver or kidney, these alkaline phosphatase level might go up. So in humans, it is involved in bone and hepatobiliary diseases. Alkaline phosphatase are also diagnostic important in different animal diseases as well. So the total serum of ALP activity has a diagnostic value, and that value is between 3 and 13 Ka units, okay, alkaline phosphatase units per 100 ml of the blood. So these are the normal serum levels of alkaline phosphatase. So if these serum levels go up and down, there could be some dysfunctioning in these organs. So then we have creatine phosphate. So creatine kinases are again isozymes of organ-specific serum enzymes, which are widely used in the clinical diagnostics. They catalyze reversible phosphorylation of creatine by ATP to form creatine phosphate. So basically, this enzyme creatine phosphatase, it will convert creatine plus ATP to creatine phosphate plus ATP. And these kinases are mostly found in heart, brain, skeletal muscle, smooth muscle, and its highest activity is found in the skeletal muscle. So in humans, creatine kinase is associated with myocardial infarction, and that is heart attack, and muscle diseases. So increasing creatine kinase in cerebrospinal fluid has been associated with a number of disorders also in animals like dog, cats, cattle, and horses. And these creatine kinase levels are sensitive indicators for muscle damage, especially smooth muscle, anything related to heart, and are largely increased in case of heart diseases. And the normal level for creatine phosphate is between 10 to 50 IU, that is units per liter, international units per liter. And if the levels go beyond this, then there is some damage with skeletal muscle or smooth muscles. Then we have alanine aminotransferases, so also known as GPT, glutamic pyruvate transaminases. These catalyzes a reversible transamination of L-alanine and 2-oxoglutarate to pyruvate and glutamate in the cytoplasm of the cell. So ALT can be found is major enzyme, and it is found in liver, skeletal muscle and heart. So the greatest specific activity is found in different animal livers. Okay. It is well established that sensitive liver specific indicator damage of, however, ALT is very low for the animal diagnostics. So it is used as an indicator of hepatopathy, that is liver dysfunctioning, or toxicological studies in which use small laboratory rodents as well as dogs. So ALT is the diagnostic marker for liver damage, and the ALT, that is SGPT levels in the serum is between 0 to 4 international units per liter. Then we have aspartate aminotransferases, also known as SGOT. It is formerly known as glutamic oxaloacetate transaminases. It catalyzes the transamination of L-aspartate and 2-oxoglutarate to oxaloacetate and glutamate. The AST is found in kidney, liver, skeletal muscle, heart, erythrocytes, and is also associated with myocardial, hepatic, parenchymal and muscle diseases in humans. So SGPT is for liver damage, and SGOT is mainly for both liver and heart related issues. The presence of AST in many tissues or serum level is a good marker for of soft tissue, but it's used as an organ specific enzyme, that is specifically for liver and heart. Red blood cells contains large amount of this SGOT enzyme, which leads to plasma before hemolysis in the blood. And the normal values of SGOT are 0 to 45 IU per liter. The next enzyme is sorbitol dehydrogenase. So this enzyme is also called L-iritol dehydrogenase, or IDH. What does it do? It catalyzes reversible oxidation of D-sorbitol to D-fructose with cofactor NAD. So the plasma activity is low in different animals, but it is found in the hepatocyte of different animals. As a result, plasma SDH is considered consistent with the hepatocyte damage. That means in the plasma, whenever the SDH is high, it is considered as a liver damage. So SDH is a liver specific for human species as well as for animal. So the increased activity can be directly correlated to the liver damage. So it's a good indicator for the hepatocellular damage in animals as well as humans. Then the enzyme is lactate dehydrogenase. So lactate dehydrogenase is the enzyme. It converts pyruvate to lactate. So pyruvate is oxidized to lactate in the presence of NAD. So lactate dehydrogenase has different isoenzymes, and this is found in heart, liver, erythrocytes, skeletal muscle, platelets and lymph nodes. So in human, it is involved in myocardial infarction, hemolysis and liver disease. So again, lactate dehydrogenase is an indicator for heart attack and liver diseases. LDH isozyme profiles were the first isozyme profiles used in clinical veterinary medicine in order to detect any specific organ damage. And the normal levels for lactate dehydrogenase are 60 to 200 IU per liter. Cholinesterases, serum cholinesterase CHE activity has major, so this we studied in the previous class also, acetylcholinesterases. So the substrate is acetylcholine, which is a neurotransmitter found at the myoneuronal junction. So acetylcholinesterase is it's a part of cholinesterases, and again it hydrolyzes acetylcholine. Okay, so which can be which acetylcholine, so that acetylcholine will no longer bind to the acetylcholine receptor at the myoneuronal junction, and it is also found in the RBCs of different animals. Only very small amount of acetylcholinesterase is found in the plasma, whereas the cholinesterase of plasma of pseudocholinesterase, butyrylcholinesterase, which hydrolyzes butyrylcholine 4 times faster than acetylcholine, is also located in the white matter of the brain, in liver and pancreas and internal mucosa. Decrease in the butyrylcholinesterases have been reported in humans with acute infection, muscular dystrophy, chronic renal diseases, pregnancy, as well as insecticide intoxication. And the normal levels for the cholinesterases are between 2.17 to 5.17 IU per ml. And then finally we move on to lipases. So there are different pancreatic lipases which are found in the serum that catalyze hydrolysis of triglycerides, preferentially at 1 and 3 position, releasing 2 fatty acids and a genetic glyceride. Lipases can be found in pancreas and hepatobiliary tract and is involved in the pancreatitis and hepatobiliary disease. And the normal levels for lipases are should be less than 150 units per liter. Again, amylases are also present in humans, and these are calcium dependent metalloenzymes that randomly catalyze the hydrolysis of complex carbohydrates like glycogen. Okay, we cannot digest starch, so it catalyzes the breakdown of glycogen, which is the major storage carbohydrate in our body, at 1,4 ligases. And the product of this action are maltoses and limit dextrins. So dextrins are the 10 to 12 glucose residues, glucose chains, straight chain glucose molecules. And the enzyme is calcium dependent metalloenzymes, which requires one of the activator ions such as chloride or bromine. Amylases can be found in salivary glands, so they are present in our salivary glands. So whenever we chew our food, we have amylases which converts the carbohydrates into sugars, and so the first digestion occurs in our mouth. Then it is also present in pancreas, ovaries, and it is used as a diagnostic aid for pancreatitis. And the normal levels for amylases are 30 to 110 units per liter. Glutamyl transferases. This is a carboxypeptidase which cleaves C-terminal glutamyl groups and transfers them to the peptides and suitable acceptors. It is speculated that GGT is associated with glutathione metabolism. The major sources are liver and kidney of glutamyl transferases, and it's also involved in hepatobiliary diseases and alcoholism. Cholestatic disorders of all the species are exampled by increasing the serum GGT levels, and the normal levels are between 0 to 30 in the serum. Trypsin is another major serum protease which hydrolyzes peptide bonds formed by lysine or arginine and other amino acids. So between lysine and arginine, it will cut the peptide bond. The pancreas has this zymogen, trypsinogen. So zymogen is the inactive form of the enzyme, and this zymogen is converted to active form, that is trypsin, by different other enzymes like enterokinase, or trypsin itself will cut trypsinogen into trypsin. Okay, and the normal levels are between 115 to 350 nanogram per mole. Then we have enzymes which are used in different immunoassays. So enzymes can be alternatives for different radioisotopes or markers. So handling radioisotopes can be quite difficult, and also it is very hazardous. So different enzymes can be used for immunoassays, can be as an alternative to these radioisotopes, and variety of proteins and can help in analysis of variety of proteins and hormones. So the role of this enzyme in immunoassay is used to replace the radioisotopes as markers. And they can be detected using more sensitive techniques. Any enzyme with sensitive and convenient assay procedure can be used for this purpose. Two common examples of enzyme immunoassay are enzyme-linked immunosorbent assay, that is ELISA, and enzyme multiplied immunotest, that is ENIT. So these are the two commonly used tests in laboratories as well as in different diagnostics. So ELISA is very commonly used for HIV detection and other virus detection. ELISA is highly sensitive and that can be used to detect antigen or antibody both. So we can check the antigen, that is virus particle present in the serum, or we can check the antibody or different bacterial infections present in the serum. Okay, so this was the end of today's outline of the 11th class, and now let's move on to the assignment question. First question is, rennet in the cheese making process separates milk into cream and skim milk, separates solid curd and liquid whey, enhances the flavor of milk, reduces the fat content. So a rennet will separate solid curd and liquid whey. Okay, so correct answer is B. The most common vegetable rennet used in cheese making is thistle. The primary role of lactose in the production of lactose-free milk is enhancing the flavor of milk, increasing the protein content of the milk, adding sweeteners to the milk, breaking down of lactose into simple forms. So lactase is generally used to break down lactose into glucose and galactose, that is its simpler forms, and thus this will help for the lactose intolerance of people in order to consume milk and dairy. The catalase in the baking industry benefits as it increases the dough elasticity, it prevents yeast fermentation, it enhances gluten formation, it neutralizes hydrogen peroxide in the dough. So it neutralizes hydrogen peroxide in the dough. So catalase will basically break down H2O2 into H2O and oxygen, and that it will release oxygen and it will neutralize H2O2. The technique used to identify various proteases in the crude cultural extract through electrophoresis is polymerase chain reaction, gelatin zymography, gel electrophoresis, chromatography. So the correct answer is gelatin zymography, B. The enzyme that converts starch to dextrine in producing corn syrup is alpha-glucosidase, cyclodextrin, glycosyl transferase, alpha-amylase, maltogenic alpha-amylase. So in plants we generally use alpha-amylase. Okay. Maltogenic alpha-amylase is generally used in baking. So the producing corn syrup, we use alpha amylase. The industrial application of maltoseric alpha amylase, again, its anti-stalling effect in baking industry, juice clarification, oxidizing glucose, breakdown of peptide. So it's used in baking, as I mentioned before, because of the anti-stalling effect. The enzyme source that produces pectinases, which are used for the removal of inner wall of the lotus seeds, garlic, almond and peanut is, so pectinases are present in the inner wall of the seeds, and the enzyme source for pectinases can be different. So the options are Fusarium, Oxysporum, Saccharomyces cerevisiae, Pseudomonas and Aspergillus species. So the correct answer is Aspergillus species. The action of proteases on protein is accelerating biological mechanism, breakdown of starch molecules, loosening muscle fibers and tenderizing the meat, improving filtration and clarification. So what proteases do? It will break down protein, starch is broken down by amylase, accelerating biological mechanism, no, loosening muscle fiber and tenderization, yeah, it will lose the muscle fibers and it will soften the meat. The enzyme that is predominantly found in papaya and used in most grocery store meat tenderizers is papain. So papain is found in papaya latex, in papaya and the tree latex. Okay, and it is again it will break down protein. Okay, and it will tenderize the meat. The reason for measuring serum levels of various enzymes in clinical setting is to monitor dietary intake, to identify cellular damage and its consequences, to regulate metabolic functions, to measure hormone levels. So we measure different serum levels of different enzymes at clinical settings is generally for the diagnostic, to identify what all organ damage or cellular damage that is occurring and what are the consequences. For example, creatine kinase is majorly present in skeletal and smooth muscle. So whenever there is a muscle damage, it will lead to increase in creatine kinase levels in serum. So at clinical settings, we can say that if we measure the blood creatine levels, so we can confirm that there is muscle damage or any organ damage. So that is why we measure different enzyme serum levels at diagnostic settings. The condition in which creatine kinase is associated in human is respiratory disorders, gastrointestinal, myocardial infarction and muscle diseases, neurological disorders. So as I mentioned before, it is used for myocardial infarction, that is heart attack, and muscle diseases. The primary neurotransmitter acted upon cholinesterases at myoneuronal junction is serotonin, acetylcholine, dopamine and GABA. So the answer is D, acetylcholine, and it is acted by acetylcholine esterase at myoneuronal junction. The mechanism that describes the inhibition of peroxidases by binding of phenoxy radicals during phenol oxidation is allosteric inhibition, competitive inhibition, non-competitive inhibition, and suicide inhibition. So the peroxide binds to the phenoxy radicals, and they irreversibly, okay, they bind irreversibly, and all this binding, allosteric, competitive and non-competitive, are reversible type of inhibition. So only irreversible type of inhibition present here is the suicide inhibition. So the correct answer is D, suicide inhibition. It's irreversible, and that is how peroxidases are inhibited by phenoxy radicals. The peroxidase is utilized in determination of glucose, uric acid and cholesterol in biological fluid, as they act as catalyst in the oxidation of these compounds. They inhibit the formation of these compounds. They catalyze reduction of these compounds. They function as coenzymes in these acids. So they catalyze. So basically, peroxidases, they will oxidize. Okay, they will catalyze oxidation of the compounds. The correct answer is A. They act as catalyst in the oxidation of these compounds. So that's all for the today's class.